Hello, and welcome to this lecture on Chapter 14. This lecture is going to cover the lymphatic system and the immune system. We do both of those because the lymphatic system is responsible for draining excess fluid from the body, and a lot of times it's in that excess fluid where you start to see pathogens, these disease-causing microbes. And so through this lymph system, this fluid is filtered, it's exposed to different white blood cells, and that's when immune reactions can occur. So we tend to pair these two systems together. So let's just look at this. Lymphatic system is technically a secondary circulatory system. Rather than transporting blood, though, it's transporting lymph fluid. Lymph vessels collect and carry away excess tissue fluid from those interstitial spaces, and that does eventually go back into the blood circulation. There are special lymph vessels called lacteals in your digestive system, in your small intestine, and these are specifically designed to transport fats. So fat does not transport easily through the bloodstream since it's mostly plasma, and so lacteals are able to transport fats more efficiently through the body. There are also other structures, things called lymph nodes, that are situated at spots along lymphatic vessels. Kind of think of these like checkpoints. And they have lymphocytes, which are a type of white blood cell to help defend against diseases. Here's just a graphic image of that. You can see your standard circulatory system, the pulmonary and systemic circulation, and then alongside of it you have this lymphatic circulation, where in the capillary beds you'll have these lymphatic capillaries right alongside your others, and these typically lie especially alongside veins. And it's this entire secondary system that's helping to transport excess fluid throughout. Because if you remember in our lecture about filtration in the circulatory system, you tend to have more fluid being pushed out than is reabsorbed. And so that excess fluid, rather than staying in the tissues, it gets absorbed into the lymphatic system. So let's talk about those lymphatic pathways, the way that the vessels are arranged. Similar to veins and arteries, they do kind of branch and get smaller and then larger. And so the smallest of these are lymphatic capillaries. These are tiny closed-ended tubular vessels going into those interstitial spaces, running alongside blood capillaries. Notice that they are closed-ended, so these do have a terminal end. They're not continuous with each other, so it's not like you go from one larger lymph vessel through a capillary bed of lymph vessels. These are just the smallest of them. So you can consider this kind of being the starting point, because it's in these where the fluid actually enters. So they receive this interstitial fluid through the thin walls, and in between the cells, we'll see that they kind of overlap. They have these layer of cells that overlap each other. And so as the fluid pressure increases, it pushes its way into inside of the lymphatic capillary. Once it is inside there, even though it's still the same thing, it's not changed by moving from the interstitial space into the lymph and lymph capillary, but its location is changing. So now we call it lymph. So that is the distinction to make. Here you can see this depiction of it. You see again those closed-ended capillaries and this one-way movement of fluid into the lymph vessel. And you can see here these overlapping cells, where as pressure will increase on the outside, it kind of pushes its way in. But then as pressure increases inside of the lymph vessel, you can see that would then force those slits closed. So that's also another way to make sure that excess fluid doesn't build up inside of the lymphatic capillaries. So lymphatic vessels, the walls of lymphatic vessels now, so capillaries to vessels, they're thinner than those of veins, still have 3 layers to them though, and they have valves on the inside like veins do to prevent backflow. Lymphatic vessels pass through those lymph nodes, and then once they do, they then merge to form lymphatic trunks. So here you can see the valve within the lymph vessel. You can see that kind of barrier of cells. Lymphatic trunks drain from lymphatic vessels and typically are named for the regions that they drain, so the cephalic, the thoracic, those kind of things. The ducts then are trunks that have been merged into ducts. There are 2 collecting ducts through the whole body, so you can kind of think capillaries to vessels, vessels to trunks, and then trunks merge into one of these 2 collecting ducts. The 2 collecting ducts are the right lymphatic duct. That basically drains the right side of the head and the arm, the right arm and kind of upper chest. And then the thoracic duct drains everything else. So it has a much larger job. It's draining from the entire left arm, the lower body, pretty much anything from the diaphragm below is all drained by the into the thoracic duct. And both of these dump into the subclavian veins. The right lymphatic duct into obviously the right subclavian, and the thoracic duct into that left subclavian vein. And if you remember from the subclavian, it can then return to the vena cava where it goes into the heart. And so there you can see that. All of these smaller capillaries, which aren't depicted, that have been merged into vessels. You pass through nodes to get into trunks. From trunks, you then merge into ducts, and then from ducts, they return into the subclavian vein. And then there's just it written out for you if you like that. All right, so let's actually talk about that tissue a little bit more. Let's talk about lymph. So that is the fluid that's inside of these lymphatic capillaries. And that formation depends on tissue fluid formation. So whether that be from water and dissolved substances that are coming out of blood capillaries, so that filtration osmosis process of fluid exiting the bloodstream and moving into the tissues, into the cells. And so this is a good chance just to kind of remind yourself that when fluid is exiting the bloodstream, it moves into that interstitial space, and then it can go into a cell. It's not like the cells are packed right up against the blood vessel. There is this space that fluid kind of moves around in cells. While some cells are anchored, and they are anchored in space, you have this fluid that things dissolve across and have to move across before it can actually get into the cell. And so that fluid, that space in there is that interstitial fluid. And so that, again, is coming from blood. It can come from waste products or products from cells. And so that fills this interstitial space. This interstitial fluid is almost the same as blood plasma. The only difference is it doesn't really have plasma proteins. Plasma proteins, your albumins and globulins, tend to be too big to pass through the capillary walls. So it does not reside in there. Those proteins, that's what creates that plasma colloid osmotic pressure, that COP, and that's what's drawing fluid back into the capillary, so reducing your tissue fluid formation. As tissue fluid formation is reduced, lymph is reduced as well. Anything that doesn't go back into the capillaries, though, that isn't pulled back in by that plasma COP, then becomes tissue fluid. And there's a minimum amount of tissue fluid that needs to exist to kind of give us the fullness that we have. When people talk about being dehydrated, and if you kind of pick your skin up, you should be able to kind of pinch your skin, and then it goes immediately back into its original shape, and kind of nice and flat and smooth. If you pinch your skin and it stays wrinkled, it's one of those signs you're dehydrated. And so there's this level of fluid that needs to stay in our interstitial space. It's once it starts to become excessive, that's when we start to have lymph formation. So filtration from plasma usually occurs to a greater extent than reabsorption. That's producing that tissue fluid. Osmotic pressure in the tissues interferes with the return of fluids to the bloodstream. So that increasing tissue fluid starts to push that lymph into the capillaries. So rather than being pushed back into the bloodstream, it gets pushed into the capillaries, the lymphatic capillaries, and that's where it's called lymph. So again, you have that filtration from blood plasma into interstitial fluid. And then from interstitial fluid, kind of the osmotic pressure, that pressure of fluid against the lymph capillaries is what pushes that excess fluid in there. So it's kind of a passive way of making sure that our bodies maintain the right interstitial fluid amount. A lot of substances, including those small proteins, are returned to blood via the lymph. So while larger proteins aren't going to be filtered out of the, or will not exit the bloodstream, smaller proteins will, and they don't belong there. They really shouldn't be in the interstitial space. They can't really be very useful there unless they're in a cell or in the bloodstream being transported somewhere. So being in lymph allows them to be transported and back into circulation at the end of the cycle. It also transports foreign particles, bacteria, viruses to those lymph nodes for recognition and destruction, and that's that immune function of the lymph system. So look at the movement of lymph. So similar to blood vessels, or rather veins specifically, lymph vessels don't have a blood pressure, so they don't have this force that's pushing fluid through them, so it relies a lot of times on other actions. So again, talking about what forms lymph, you have the hydrostatic pressure of tissue fluid. Interstitial fluid builds up and it pushes into lymphatic capillaries. And then once fluid is in lymphatic capillaries, once you have that lymph movement, it basically relies on the skeletal muscle contraction, breathing movements. We saw both of these in the venous system as well. Contraction of smooth muscle in the walls of those lymph vessels, and then the valves to prevent backflow. So very similar to veins. What can happen is called edema. This might have been something that you've heard of before or experienced yourself or know people that have. And this is when you have an accumulation of lymph in those interstitial spaces because there's some interruption with the flow of lymph. So this is when that basically it's saying that fluid is not being drained out of the interstitial spaces and it's allowed to be collect there instead of moving through the lymph system and being into recirculation. And so when that happens, you start to get swelling. This example during surgery, removing cancer breasts, lymphatic tissues or vessels may be removed or disrupted. Obstruction results in edema. So you're having surgery. This is a planned thing, but of course, when they're in there, because it's part of the lymph system, this cancer that we're referring to in this example, they remove some of those vessels, and so that then interrupts the flow. It stops some of the flow, and so there's going to be edema. So their doctor might prescribe meds to help reduce inflammation or reduce fluid collection, maybe compression garments to help just add that physical compression of squeezing and pushing the fluid through, maybe massage, multiple things that they could do to help counteract this edema. If it needed to be drainage, they would put in a drainage tube to help with that. Okay, so now let's look at some more lymphatic tissues and organs, because lymph system is not just those lymph vessels. As we talked about, we mentioned lymph nodes a couple of times. So these lymphatic tissues, which lymph nodes are a type of lymphatic tissue, they have lymphocytes, macrophages, and other cells in there acting in this filtering job. There's two types. There's unencapsulated and encapsulated lymphatic organs. Unencapsulated lymphatic tissue, those are things like your tonsils in the back of your throat, the back of the nasal cavity. Those are technically just big clumps of lymph nodes that don't have this formal capsule around them, kind of collecting them into an organ shape. Because a lot of times when we think about organs, what we're really seeing is just an encapsulated version that's, we think of as the organ, but really an organ at its functional unit is much, much smaller. Same kind of thing here. This is, an aggregate of lymph nodes that just isn't covered up, finished wrapping. But that also helps it with its function, because if you think about having lymph nodes, unencapsulated lymph nodes in your nasal cavity or oral cavity, this allows any pathogens to make direct contact or much easier contact with the lymph node, rather than having to go through layers and layers and layers of connective tissue covering it. These can have a lot more direct contact, much more easily identifiable, especially in these places that are so susceptible to infection. Your tonsils, your appendix, Peyer's patches, and the intestines. These are all places where are rife for pathogens to be able to enter the body. And so having these lymph structures in there is very helpful from that immune function sense. The others are the encapsulated lymphatic organs, lymph nodes, thymus, and spleen. So we'll look at these specifically and kind of what they do and their functions. Starting with those lymph nodes are groups located in groups or chains along lymphatic vessels. So if you ever go to the doctor and they're doing all their assessment of you and they're listening to your heartbeat and checking your pulse and your temperature and all those things, and then the doctor comes in and they start to kind of palpate and they kind of poke around behind your ears and they touch along your neck and maybe under your jaw a little bit, maybe even into your armpit sometimes. What they're doing is palpating your lymph nodes. They're palpating to see if your aggregate these collections of lymph nodes are swollen or not, because swollen lymph nodes is usually a big sign that your body is fighting some type of infection. So you could still be feeling well, or at least what you consider to be normal, and fighting an infection at the same time. Or maybe it's a sign of some circulatory issue or heart issue or cancer that you might be fighting. There's lots of things that could be happening in your lymph system. And so just by doing that just straightforward palpation into the lymph nodes, it can be somewhat diagnostic and help with the doctors. So lymph nodes are very small. They're these little bean-shaped nodules. They have blood vessels, nerves, and vessels entering or exiting at what they call the hilum. So we've seen this structure many times before. So again, these are covered in connective tissue wrapping, so they are very much sealed organs, and so they have to have some type of entrance exit, and that's through the hilum. Afferent vessels arrive, so A arriving, E exiting, just remembering that. Typically you have multiple afferent and one efferent vessel. They're covered with that connective tissue capsule. It extends inside of the lymph node to create lymphatic nodules and sinuses, those little open spaces, so that lymph can be further divided. It can be split up, because when you're just looking at one big mass of fluid, it's hard to really see if there's any pathogens in there. But once you start to split it up into 5 or 6 different little, smaller pools, it's a lot easier to filter through and sift through. So lymph enters through the afferent vessel. It gets divided into all these lymphatic nodules for filtration, and then it exits through the efferent vessels. Lymph nodes have both lymphocytes and macrophages, two different types of leukocytes. Lymphocytes being your B cells and your T cells, and macrophages are a type of monocyte. They're there as a phagocytic cell. They are what filters lymph as it flows through them, removing a lot of pathogens, macrophages, through phagocytosis, right? That's in their name. They're consuming and eating these cells, whereas lymphocytes have more specific responses we'll look at. That filtered lymph then exits through the efferent vessels. Here's a nice little depiction of that. You can see these afferent vessels arriving. This one, or I guess technically two, these efferent vessels that are leaving. And then, of course, because this is an organ, it has its own blood supply to help make sure that it's staying nourished, that it's getting fresh leukocytes for immune filtration. There you can also see a lymph node. That's the lymph node right there, and then it has its lymphatic vessels all around it, and blood vessels too. Lymph nodes generally occur in chains along parts of the larger lymphatic vessels, so lymphatic vessels being the things that bring lymph to the node, the trunks are what take it away. Not found in the central nervous system. Most concentrations are cervical, thoracic, axillary, supratrochlear, abdominal, pelvic, and inguinal regions. I would expect you to know pretty much all of these, like their locations. The one that might be a little tricky is the supratrochlear. That's along the elbow. And so that's the only one that I can think of that might not be kind of come right to your mind. There it is. You can just kind of see, again, the supratrochlear ones, cervical, thoracic, axillary. So these larger collections of them. There can still be other lymph nodes scattered all throughout your body, though. These are just the larger collections that we tend to find. So the main functions of lymph nodes, they are there to filter lymph, removing bacteria and cellular debris. They act as immune surveillance, monitoring body fluids. The lymphocytes and macrophages help to do that. They're centers of lymphocyte production. So we'll see that once lymphocytes, if they do recognize any pathogens, they can replicate within the lymph nodes. Inside of a lymph node, this is where lymphocytes are also able to actually attack viruses. And then you have the macrophages engulfing and destroying foreign particles, any damaged cells, anything that's kind of just floating in the lymph that needs to be filtered out. So looking at the thymus now, another one of these lymphatic organs. The thymus is this kind of soft organ that sits just behind the sternum on top of the heart. Not really on top of the heart, but kind of in that general area. Anterior to the aorta, right, just behind the sternum. You can kind of think about it in that terms. It starts to shrink in size during our lifetime. It starts off larger when we were children, and then by the time we're adults, it's mostly adipose and connective tissue. So it's not really as functional as it is when we're in children. It too is divided into lobules, just like the lymph nodes are. And those lobules have lymphocytes. But what's interesting in the thymus is that these really aren't for filtration purposes. It's not like blood is flowing through the thymus to be filtered. The lymphocytes in these are being matured into T cells or T lymphocytes, same kind of thing. I'll use that interchangeably, T cells and T lymphocytes, and once we learn B cells and B lymphocytes, it's the same things. So the thymus, that's where the T comes from. Thymus are T cells, they are immune cells, lymphocytes that have been matured in the thymus. The thymus also secretes thymosins that influence the maturation of those T lymphocytes. There you can see where the thymus kind of sits. Again, just supero anterior to the heart. And then the spleen. This is the largest of your lymphatic organs in the body. It sits on the kind of upper left quadrant of the abdomen, just kind of behind the pancreas and the stomach, off to the side. A lot of times when people think about the pancreas, they think about it just as a blood reservoir, which that is one of its functions. It does house a lot of red blood cells, and so that is something that it can do during times of blood loss. It will start to release those blood cells to help re-up our blood levels, but it also provides this filtration process. While lymph nodes are going to be filtering lymph, the spleen does filter blood. It has blood flowing through it, and that's what it's filtering for its role. It does has both white pulp and red pulp. White pulp rather, is mostly lymphocytes. So you can think lymphocytes, leukocytes, white blood cells, white pulp, and then red pulp, which has mostly red blood cells. So those two can kind of, that's why they look like that. It also has some macrophages and lymphocytes. The spleen is mostly responsible for filtering blood of pathogens and removing any damaged old red blood cells, and like I said, being a reservoir for excess blood. There you can see, kind of sitting off, this kind of concave shaped organ. It's different red pulp and central white pulps, and you can see the vessels kind of flowing through it, how they're divided into these different sections. Okay, so let's get into actually the body's defenses against infection. We'll start with talking about what pathogens are, and then getting into some of the different reactions that our body has, or strategies our body has for dealing with infections. So starting off with the word pathogens. A pathogen is a disease-causing agent. This can be a bacteria like E. coli. It could be a virus like the influenza virus. It could be pathogens like intestinal parasites, things like that. These are all different types of things that can cause disease, cause illness in the body. And so our body has multiple ways to protect against this. Let's see. The two basic mechanisms are the innate and the adaptive defense, or innate and a non-specific and specific immunity. Innate means that it's something that you're born with. This is something that is just innate to our species. So as humans, we can expect that everyone is going to have at least these basic characteristics to provide immune function. Some people will have more robust and less robust immune systems, but for the most part, we are all going to have these same things. When someone does have a less robust immune system, or if they are compromised in some way, they're lacking certain aspects of this innate or adaptive immune system, that's when we might say that they are immune compromised. So their body's natural defenses aren't as varied and rigorous maybe as others could be. And so that's something we're going to look at, what these different defense systems are. So the innate, your non-specific defense system, guards against many types of pathogens. It is, again, non-specific, so it doesn't have to be a certain kind, like bacteria or virus. It doesn't matter if it's a specific bacteria down to the actual species of it. It's just going to respond really quickly, very generically. This is stuff like mechanical barriers, your skin, chemical barriers, natural killer cells, the inflammation process, phagocytosis, fever, all these kind of general processes our body gets into. The adaptive is more specific, so it's something that your body learns and gears towards the specific pathogens you're exposed to. It's one of those reasons why, when children are really sick, kids get sick all the time. They just get sick all the time because they're being exposed to a lot of new things that their body's never seen before. They get some immunity from their parents, especially through breastfeeding. That provides that kind of passive immunity of allowing antibodies to help fight infections during those first few months. But then, as they age, it really just comes about exposure, making sure, that healthy limit between, okay, let's, get out there, and kids are going to touch things and put everything in their mouth, because that's the best way to tell if something is good or bad or not, is to put it in your mouth. And so they are always getting sick. They're always getting exposed to these pathogens. And so their body is having to catch up. So it's adapting to the things that they're being exposed to. But then as an adult, you don't get as sick as much. One, you learn, okay, don't put things in your mouth. And two, you probably aren't being exposed to as many new environments. Maybe if you go travel somewhere, and let's say, especially if it's typically recommended to get vaccines or take antibiotics for certain things prophylactically before you travel somewhere, and you don't, yeah, you might get sick because you're getting exposed to all these new pathogens. And so that's because your body has not adapted to this yet. That's what typically happens in a lot of colonizer situations, is that groups move in, they're bringing these new diseases that they already have defenses against, but the people that they're encountering do not. And so it, can be true epidemics that can be spread. Adaptive immunity, a lot of times it is more slowly to respond because it's responding to specific pathogens and it has to be activated through certain steps. It uses lymphocytes. Those B and T cells are going to be secreting things called cytokines or potentially antibodies. So species resistance. This is a species resistance disease that affects other species. So some things that we have just natural resistance to that maybe, you know, dogs or cats, it could be deadly for them. This can be different chemical environments. So some of the chemicals that we produce are different from the ones that a dog might produce. And so, yeah, we're gonna, like in our intestines, in our digestive tract, we might have, you know, a different pH that does not allow for bacterial reproduction that maybe would cause infection in another animal. Body temperature. Smaller animals tend to run slightly higher in their body temps. It's normal for a dog to be up to 102, and so they're going to shift their profile of pathogens. And then just absence of receptors for a particular type of pathogen. This kind of think about this in terms of like blood cell transfusion. So remembering our ABO and RH blood groups, how every cell has a receptor on it, a flag that identifies whether, you know, it is a body's normal natural cell or whether it comes from something else. And so if you don't have a receptor for certain things, or if you don't have a way to detect for certain things, or a way for a pathogen to bind into your body cells, then yeah, it's not going to cause any infections. Mechanical barriers. So mechanical barriers are preventing entry into the body. Your mucous membranes in your mouth, your nasal cavity, your genital tracts, those are places that are susceptible for infection. So we have lymph nodes there. We have those MALT, those Pyre patches to make sure that any pathogens can be filtered through. There is just your skin. Unbroken skin is a really good way to protect just generally against pathogens. That's why when you have scratches and scrapes and cuts and things like that, it's always good to make sure, you know, you're monitoring them. What color are they? Are they really swelling and hot? Do they need to be covered up? So that way you don't have any pathogens getting into those places. Mechanical barriers, we call this the first line of defense of the body. So your mucous membranes and your skin, first line of defense of the body. Second line of defense is the rest of these innate defenses. So inflammation, this is part of that second line. So let's say something does get into your body. Well, your body's going to respond. It can respond one way through inflammation. That's a localized response. Wherever your cut is, maybe that spot starts to get red and swollen and really warm and hot. And so that is really actually a way to help stop the spread of pathogens. It's allowing the blood vessels to dilate to increase the flow of blood volume to an area. That allows for white blood cells, neutrophils, things like that to be brought into this space to be able to filter through and defeat and eat up any pathogens that might be in the area. It's also increasing the heat. Some bacteria, some pathogens do not do as well if we increase the heat a little bit. It actually makes macrophages more active when the body heat goes up a little bit. And so those things, just again, the different physiological reactions to inflammation are a part of their immune defense reaction. So it's bringing all these fresh blood cells, bringing fibrin, bringing platelets into the area that's allowing it to be kind of flushed with these nice new nutrient healing cells. And what do you know? When you have a bunch of fluid moving into an area, it's going to increase the pressure within that interstitial space, and it's going to push that excess fluid into the lymph system, where instead of spreading to potentially other organs, it can then, and back into the bloodstream, it can be filtered through the lymph system and filtered through lymph nodes to be able to, once again, make sure there's no excess pathogens in that space. Chemical barriers, this is, so rather than like a macrophage eating it or a mechanical barrier stopping it, this is things like our acidic environment that could start to denature the proteins of certain viruses or bacteria. Enzymes like pepsin and lysozymes and tears to help destroy pathogens. Things like interferons, which are hormone-like proteins that essentially can block viral replication. They slow tumor growth. They interrupt cell processes. And then complement. Complement is a group of plasma proteins and fluids. It stimulates inflammation. That helps to do all those processes that we said inflammation can can accomplish. Complement can be pretty upsetting. That's one of the things when people were having large reactions to COVID in the beginning. A lot of times we were talking about this complement cascade, where they were having all of this inflammation in their lungs because the complement was going there trying to stop the virus from spreading, but it was causing this overreaction. Natural killer cells, NK cells, these are a type of lymphocytes, not your B or T cells, a different kind. And they fight against pathogens by secreting these substances called perforins. And if you think perforin, perforate, what they do is they kind of poke holes in the cell membrane so that it kind of lyses up and just breaks open. They also can secrete substances to help it with inflammation. But again, very general, just kind of, oh, yep, you're not a normal body cell. Let's just go ahead and get rid of you, secreting these perforins. And then phagocytosis. This is accomplished by a couple of different types of cells. It's when you're engulfing and digesting pathogens, foreign particles, debris, because debris, little cellular debris, that can be a way for some pathogens to get nutrients or to have the resources they need to replicate. And so keeping things clean and tidy, make sure that that doesn't happen. The most active of phagocytes are your neutrophils and your monocytes. The monocytes will actually leave the body through that leave the bloodstream through that diapedesis and kind of rest in tissues, and then they become macrophages and kind of can be specialized to that tissue area. Phagocytes are attracted to an area by chemotaxis. This is when infected cells, damaged cells will release chemicals to help draw those phagocytes directly to them so they know where to go. Neutrophils typically for smaller particles, monocytes for larger ones. Again, monocytes, once they exit the body, or exit the bloodstream, they become macrophages, either kind of roaming in that tissue space, or they might just kind of hang out in one spot and say, this is a good spot for me. I'm going to sit right here and just watch this corner. And macrophages function great. Neutrophils plus monocytes plus macrophages equals your mononuclear phagocytic system, or your reticuloendothelial system, which is probably just a lovely little piece of knowledge that hopefully will come in handy. All right, so fever. So we talked about inflammation being a localized response. Fever is an all-over response in the body. Fever occurs when the body's temperature is set to a higher point. So rather than trying to bring it back down to its set point in the hypothalamus is actually raising your body temperature and holding it there. It makes a hostile environment for pathogens, so it can, normal pathogens that are going to affect us and make us sick. If we raise the temperature, sometimes just a couple of degrees is all that it takes. That is enough for them to become sluggish, to become slow, to not function quite as well, or potentially to denature and die. The elevated temperature also causes the liver and spleen to take up iron, reducing that amount in the blood, and which ultimately takes away resources from pathogens. And phagocytic cells, as I said, attack with more oomph when the temperature is up a little bit. When they're feeling a little warm, they have a little bit better activity. So that's why usually it's said if the fever is mild, if it's not too bad, which typically 101, 102, once it starts to get up to 103, 104, 105, that's when you really have to worry. 105, I believe it's 105 is the threshold for brain damage. So if it gets to that point, you're probably starting to get dehydrated. It's not just a fever thing. Your body is starting to not have as enough water to be able to keep itself cool. So always balancing your overall safety with, all right, if I can ride this fever out, it is doing something to fight an infection. Okay, now let's look at the adaptive immune system, the specific immune system. So whereas the innate immune system, like I said, was just very general against all kinds of pathogens, everybody's going to have, for the most part, the same innate immune system. The adaptive immune system is going to be unique to each individual, even though the overall structure is the same. So immunity is a response by the body against specific pathogens, their toxins, or metabolic products. So response by the body against specific pathogens, so a specific bacteria or virus that your body has seen before. Their toxins, so maybe a parasite is secreting toxins and your body has a way to counter it, or their metabolic products. Again, maybe something is being released into your bloodstream that your body is able to counter and isolate and neutralize. This is the third line of defense against pathogens. So if they can't be stopped from entering the body, if they can't be neutralized with a fever or with a macrophage, and they are able to infect your body cells and actually cause an immune response, this is the immune response. This is that third line. So this is typically performed by lymphocytes and some macrophages to help recognize and remember specific foreign molecules on particular pathogens. We call it your cellular immune response and the humoral immune response. So there are two different kinds. Within adaptive immunity, the specific immunity, you have both cellular and humoral responses that we're going to kind of examine and look through. So here's a little flow chart of that if you want to. Pathogen attempting to gain entrance to the body. Your first line of defense is mechanical barriers. If they can get through that, the pathogen enters the body. It then has to deal with the second line of defense. And while it's going through this, it may activate our third line of defense as well. So first starting with antigens. So antigens are molecules that can evoke an immune response. Remember, if someone has blood type A, that means that they have the A antigen on their red blood cell. And if you give that to somebody that has type B blood, their blood will recognize the antigen on that red blood cell and attack it. And so antigens are these molecules, these flags, right, that kind of stick onto cell surfaces to help identify them. They can be proteins, polysaccharides, glycoproteins, glycolipids, all kinds of different types of structures. Before birth, the body makes an inventory of the self-antigens. So it says, okay, these are the range of self-antigens that are normal to us, and we're not going to attack those. If we see these on a cell, we know that it's normal and that it's safe. Anything else, we can go at it fair game. So the immune response is directed against these non-self molecules. They tend to be large, complex, foreign molecules in the body. Sometimes smaller ones called haptens will join with larger molecules to become antigenic. This is a lot of times how food allergies occur, is that you have a small hapten, a small protein that joins with a larger one, and then it kind of amplifies that response. So speaking of lymphocyte origins, so we said that the adaptive immune system is based off of mostly lymphocytes, the B and the T cells. And so that starts in red bone marrow. Red bone marrow releases what we call undifferentiated lymphocytes into circulation, starting in fetal development and continuing throughout the lifespan. We call them undifferentiated because once they go through that maturation process, they'll turn into B cells or T cells, specific lymphocytes. Lymphocytes come from that undifferentiated hemopoietic stem cells. Half go to the thymus, where they become T cells. They make up the majority of circulating lymphocytes, and then some will settle in the lymph nodes, the spleen, and the thoracic duct. So these lymph structures, where again a lot of lymph fluid or blood is being filtered through. So T cells mostly go into circulation, with some hanging out in the lymph organs. The rest become B cells. B lymphocytes are B cells. I say B cells, it's just easier. The B cells, there's again kind of basically flipped. 20 to 30% of circulating lymphocytes, and then the rest settle in those lymphatic organs for monitoring to see if they recognize any pathogens. Here is a B cell, just a typical B cell. So here, take a moment to read through the steps of production of B cells and T cells in the bone marrow. Starts in the bone marrow, that gets transported through the blood to the thymus, where they become mature T cells, and then the others just circulate through the blood or they settle in lymph organs, whereas the B cells stay in the bone marrow, mature there, and then they're released into the bloodstream, where the majority rest in lymph organs. Here's a nice little table just to be able to help you understand that. Okay, so let's look at the T cells and their cellular immune response. Sorry, I had to get a little water. So lymphocytes require activation before they respond to antigens. It's not just like T cells and B cells are constantly being active. They have to have sometimes a couple of signals before they initiate their immune response. So T cell activation requires encounter with an antigen-presenting cell, an APC. And this is essentially a B cell or a macrophage, something that has encountered the pathogen and can then tell the T cell, hey, this thing's in here and it's dangerous. T cells can't recognize dangerous antigens on their own. They can't just bind to a pathogen and say, oh yeah, you're not good. They have to have it presented to them in some other way. And so that's the first step in activation, is that T cells interact with an antigen presenting cell. So macrophages as androgen APCs, I'm just going to say APCs. Macrophages as APCs, essentially what they do is that they eat the pathogen, whatever disease-causing organism it is. They engulf it and draw it in, and then once it's inside the macrophage, they break the pathogen down, and then they take some of the proteins from that pathogen and they combine it with their own normal body protein, with their own normal cell proteins, and they put that on their surface. And it's called this major histocompatibility complex. Histocompatibility, tissue compatibility, right? So this kind of idea that this is a matching thing for us, that this matches our body. So the MHC, the major histocompatibility complex, is going to be mixed with these pathogen proteins, and so that tells the T cell, oh hey, this is not right. Right? It can read the MHC. The MHC kind of puts it into a format that the T cell can recognize, and the T cells are going to be able to bind to that, and that's what allows them to recognize those displayed antigens. Once they recognize and bind to those fragments, they match their receptors, and this can take multiple hours. It can take several hours for this process to happen. They then become activated, and that's good because you don't want your T cells to be too easily activated. Otherwise, you can start to go into like autoimmune responses. So this is the cellular immune response, or cell-mediated immunity. It's both the same things. The response directly through cell-to-cell contact, as activated T cells interact directly with antigen-bearing cells. So the cellular immune, the cell-to-cell contact it's talking about, is between a T cell and an APC, like a macrophage or a B cell, not cell-to-cell, meaning T cell and pathogen. Those two things do not directly interact. Except that the T cells are going to secrete toxins to destroy those pathogens. So T cells also synthesize cytokines to enhance cellular responses to antigens. So they kind of act as this helper role to help kind of push along the immune response. And some cells secrete toxins, growth inhibiting factors, interferon to help destroy those pathogens. CSFs, your colony stimulating factors, are cytokines, a type of protein, stimulating white blood cell production in bone marrow and activating macrophages. So if again, it takes a couple of hours for this cell-to-cell contact to really activate T cells, because there is this big response that can follow depending on how severe the reaction. So the types of T cells, like I said, T cells have a variety of functions. There are helper T cells, cytotoxic T cells, and memory T cells. Helper T cells are going to stimulate B cells to produce antibodies against the displayed antigen. They also, we'll see, kind of secrete some other hormones to help create memory cells. Cytotoxic T cells, cytotoxic, so this that name should give you an idea that they are going to be destroying other cells. They monitor the body cells, recognize cancer cells, and virus-infected cells. They do things like secreting perforins to help, again, lyse these cells that have been infected. Cytokines from the helper T cells are going to activate these cytotoxic T cells, and then that can also cause them to, through their activation, start to replicate and produce even more. Through that replication, this cytotoxic T cell can also create memory T cells. They create a quick response to any future exposure by quickly turning into, dividing and turning into a lot of cytotoxic T cells. So here, I know this looks confusing, but again, take some time to go through it because you have a lot of concurrent things happening, right? So starting at level 1, macrophage is phagocytizing and eating this antigen. So here's your pathogen with its antigen markers on its cell surface. The macrophage is going to engulf that, break it down, and then bind some of these antigens to the MHC for displaying. And that's going to allow the cytotoxic T cell to recognize it. It's going to bind there. You're having the cell-to-cell contact. You also have this helper C cell over here, kind of allowing it to be it to be activated as well. So cytotoxic T cells, helper T cells, both being activated. This cytotoxic T, or rather, let's start with the helper. The helper T cell is going to proliferate. It's going to start to divide and make more of itself. And then you can see it releases interleukin, which helps to further activate the cytotoxic T cell to become more of its cytotoxic self and to produce memory T cells for future responses. This is how cells, why you can encounter a pathogen one time and it might make you really sick, and the second time you barely get a sniffle because your body has all these memory cells to be able to remember these infections and remember how to fight it. At the same time that your T cells are being activated, your B cells can be activated. They can bind to antigens on a pathogen, and they make that direct contact. So your B cell is being activated here, and then once it has these cytokines from the helper T cell, so cytokines for B cells, interleukins for T cells, the helper T cell releases those cytokines, and this can be the same cell. You can have one helper T cell releasing cytokines for B cells, and the same helper T cell releasing interleukins. Those cytokines, though, will activate the B cell, and we'll see it go through its process of differentiation and antibodies. Here we go. So let's get into B cells. So B cells in the humoral immune response. So cytotoxic or T cells were your cell mediated, whereas B cells are humoral immunity mediated. Humoral refers to the blood. It refers to this ability of things to be able to move through the bloodstream. So this one does not require an antigen presenting cell. A B cell can be activated and do all of its full process through binding of the antigen on a pathogen. But most of them need helper T cells for activation as well, which is one of the nice ways that B cells can activate those cells. If a B cell, binds to a pathogen, it can then notify a T cell before the T cell has even seen it. So the B cell can kind of say, "Hey, catch up, hurry up! I need some cytokines to be able to actually do what I need to do." And the helper T cell will do that. It'll encounter that B cell, release its cytokines, and then activate and stimulate that B cell to proliferate. Some of the B cells become plasma cells, and those eventually are what produce antibodies. So B cells don't produce antibodies. B cells turn into plasma cells, and then plasma cells produce antibodies. Antibodies are immunoglobulins. So globulin as those plasma proteins, immuno for the immune response, your Igs, right? Antibodies travel through the body fluids to destroy and attack antigens on the pathogen surface. So that's why they call it the humoral immune response. Other B cells become memory B cells. They stay dormant. Memory cells, whether they're B or T cells, are not actively fighting in infections. They are just there to kind of say, "Oh, hey, we've seen this thing before. Oh, let's divide and make some of our active versions of ourselves, and maybe a couple of more memory cells because this thing's come back once, it'll probably come back again." And so that's the way that our body kind of keeps memory fresh for immune memory. Here's the process of B cells being activated. Again, this antigen presentation onto the B cell, cytokines from a helper T cell. It's going to start to divide, and then you have plasma cells secreting antibodies and memory cells which remain dormant through the process. Here are those same steps just kind of written out, so you can read through those if you want to see that for B cells and for T cells, and to try to compare how they're working side by side. And then we can look at antibodies. So the specific antibodies that are produced by B cells. There are 5 major types, but there are others. 5 major types of antibodies. They are the gamma globulin fraction of the plasma, the most abundant being IgG, IgA, and IgM. So IgG is found in tissue and plasma, defends against bacterial cells, viruses, toxins, and activates that complement cascade. IgA, exocrine gland secretions, breast milk, saliva, tears, intestinal juices, bile, urine. IgM, found for food antigens and bacteria present, react with certain red blood cells during transfusions or mismatched blood. IgD is it's found on the surface of most B lymphocytes and helps with B cell activation. And IgE, basophils and mast cells associated with allergic reactions, parasitic infections, things like that. So it's helpful to know kind of generally when you see, let's say you're doing have an infection and you're doing a blood analysis, and you see that somebody has a lot of IgG, that can give you an idea that, oh, okay, maybe they have, a bacterial infection based on other information that you've been given. And so you can find out the specific antibody that's been activated to help narrow down what type of infection you're dealing with. Three methods by which antibodies react to antigens: direct attack, activation of complement, or local inflammation. So they can do a direct attack through things like agglutination, precipitation, neutralization of antigens, just making it more susceptible to phagocytosis. We'll get into those. We'll get into all these just a little bit more. Activation of complement results in opsonization, which is a fun word. Opsonization, chemotaxis of white blood cells, inflammation process, of course, agglutination, neutralization, alteration, or lysis of antigens or antigen-bearing cells. So complement plays a big role in being able to neutralize and isolate pathogens, as well as stimulation of local inflammation. So antibodies can stimulate local inflammation through inflammatory responses and stimulating certain cells to release things like heparin and histamine. So actions of the complement system, rather. So complement system is just kind of hanging out, doing its own thing, and then presence of antibodies combined with antigens is going to stimulate this to be activated. Once it starts to get activated, it does things like opsonization. So opsonization, maybe you can think optimize. So it enhances phagocytosis of antigens, basically by the antibodies clumping around this pathogen. It allows the phagocytes to make it more obvious. It's like, oh, there it is. I can see it a little bit easier now. And so it optimizes that process. Chemotaxis, attracting lymphocytes or leukocytes rather. Lysis, rupturing cell membranes, so complement can lyse cell membranes. Agglutination, you remember that, it's clumping of antigens, so the complement proteins will bind to the antigens to make them kind of precipitate out and be more, again, noticeable. And neutralization. It can alter somehow the structure of the virus. It can change actions on its cell membrane or receptors on its cell membrane so that the virus can't bind with other cells. So through this release of antibodies and of B and T cells, our bodies can have we can see a difference between the first and second time we're exposed to a pathogen. Again, that first immune response tends to be a little bit slower. We tend to get a little bit more sicker, have more symptoms, whereas the second time it's not so much. So the primary immune response involves the activation of B or T cells during that first encounter with an antigen. They appear in the plasma 5 to 10 days after exposure. So it can take, up to, a week and a half or so of being sick with something before your body, is really starting to fight and you're starting to feel better, or even longer, 2 weeks sometimes. Because if the antibodies aren't appearing until 10 days after exposure, it can take a while for those things to still kick in. Some antibodies released into the blood to help destroy antigens for several weeks. So it's that thing of don't stop taking your antibiotics until they run out. Don't just stop taking them when you feel better, because you can have a low enough level of pathogen to still be infectious and to still be susceptible to an infection, but not actually experiencing symptoms. And so it's that low level of infection that your body kind of leaves antibodies circulating for weeks, and why you have typically antibiotic prescriptions are 2 weeks in duration, if not longer. It allows for kind of a continuous monitoring for a couple of weeks to make sure that nothing comes back up. And then, of course, some B cells stay as memory B cells. Same thing with T cells. They stay as memory T cells, remaining dormant, but then remembering the antigen in case they need to reactivate. This is why you sometimes get boosters for certain vaccines. So things like your COVID and your flu viruses, your flu vaccines, you get those every year, a couple of times a year, because your body does not, one, the memory cells don't last as long, and because the strains change so much. With COVID and with the influenza vaccines, they can actually adapt to the most dominant strains at that time to give you an even more kind of specific or targeted immune response. But then something like your tetanus tetanus vaccine. If you get a tetanus booster, it's good for 10 years. If you get your hepatitis booster, your hepatitis vaccines, those are good for your lifetime. So don't take that as medical advice, by the way. If your doctor tells you to get these boosters and get these things, go get them. But generally, some memory cells can last for 60, 70, 90 years. Maybe not that long, but you know what I mean. The secondary immune response then, so after the first one that can take a couple of weeks to really get active, the secondary immune response tends to be rapid, longer lasting, and is much more vigorous in its response. Memory B cells proliferate, antibodies appear in 1 to 2 days versus 5 to 10 days, and they remain for months to years. Memory B cells, like I said, can live for many years, providing long-term immunity for something. So this is why, again, with those first rounds of COVID vaccines, we were all instructed to get one round and then wait a few months. So wait until this first reaction had both peaked and then driven off, and then you get a second one, and you get a second dose, a second hit of it, and your body has an even bigger reaction to it. And that's what really kind of helps it to keep this long-term immunity. And then you get boosters along the way to help keep you updated for the most recent strains that are being passed around. So here you can see a depiction of that primary versus secondary response. Going up and down, you have the antibody concentration. So the first time you might produce, 25% or so, but up here it's kind of the 80-90% production of antibodies, and again, much longer sustained response. So now let's look at the different types of immunity. We had our immune responses. We talked about adaptive versus innate, but this is specifically referring to our body's immune reactions. So immunity can be classified by how it's obtained. Naturally acquired or artificially acquired. Naturally acquired is when somebody coughs on you, you're taking the bus and somebody coughs on you in your face and you're like, well, great, I just got sick. Or, you go and lick a handrail and you just, natural events, you get sick that way. Artificially acquired is by getting an injection, by a vaccine or something. It can also be classified as active or passive, and this refers to whether or not you have memory cell production happening, whether some memory B cells get produced or not. During active immunity, your body is exposed to this pathogen, exposed to this antigen, and it makes these memory B cells. During passive immunity, you are receiving the antibodies that the B cells would be producing, and so you're not actually making your own. And so you have short-term immunity, but no real long-term, no memory cells produced. So look at these kind of different combinations that we can have. Naturally acquired active. This is, again, someone gets you sick. I was raised before the chickenpox vaccine was available, and so when my sister got chickenpox, my mom put me right there beside her for a couple hours, and then what do you know, I got chickenpox too. It's not a great way to do things because chickenpox, once you have that virus, then of course you can be susceptible to shingles later on. It can be pretty dangerous, if it's a really large reaction. So we don't do that anymore, and now we have chickenpox vaccines. But back then, that was what you did. You just exposed your kid to whoever already had it, so that way they could go ahead and get it through it, and not have to worry about it again. Because it provides active immunity. By being exposed physically to this virus and by actually getting sick myself, my body was forced to create the antibodies for it, and so I have this naturally acquired active immunity. The other kind is artificially acquired active immunity. This is, again, something like with vaccines, flu vaccines and COVID vaccines. I don't love using that one because it is important to get boosters for the new strains that come up. But it is one of those examples where, yes, you are being given a weakened or deadened version of this pathogen, but it's still just enough to cause your body to trigger this immune response. So you're getting an injection. You're not actually getting sick. A lot of times people will say, "Oh, I got the flu vaccine and it gave me the flu worse than anything else." It's like, well, no, maybe you were already getting sick, and then because you had this vaccine and your body was primed for an immune response, you had a larger reaction. Maybe it was psychosomatic. You just thought yourself into that immune response. You're not actually getting an injection of the flu. It's very much a modified version of it. Same thing with COVID. You're not getting an injection of COVID. Those immune responses are separate from the actual disease that it's trying to prevent. But what happens when you get those vaccines is that your body is being exposed to a modified version of this pathogen so that it's able to examine it from a safe perspective. It's able to initiate an immune response, figure out what it needs to do, make memory B cells, and then it's ready in case you ever do get infected. So vaccines do not stop you from getting infected. You can still get an infection when you have a vaccine. It's kind of like how a contract doesn't stop you from getting sued. Like you can you can have a contract, but somebody can still sue you, and that just means that they're taking you to court to make sure that you followed the terms of the contract. So contracts don't stop you from getting sued. You can still get sued. Vaccines don't stop you from getting infected. You can still get infected, but they protect you from something much, much worse. You may even still get a little sick. You might still have to, maybe you did break that contract a little bit and you have to pay a little bit of money. It's better than having to pay millions to somebody. Same thing with a vaccine. It's better to just get, like a little headachy, a little tired for a day or two compared to being hospitalized and on a ventilator because you can't breathe. So artificially acquired active vaccines that your body can then produce its own antibodies. It's being exposed to the virus or bacteria. Naturally acquired passive. So a naturally acquired passive immunity, this is meaning that you are not producing memory cells. So passive immunity, no memory cells are being produced. This is through breastfeeding. Typically, that's how that happens. During those first 6 months or so of life, infants aren't really producing enough antibodies themselves. And so through breastfeeding, they are just getting the antibodies that they need to fight the pathogens that are in their general environment. Because again, they're getting them typically from the mother. And so whatever the mother's being exposed to, the fetus or the infant is being exposed to as well, most likely. And so it's kind of a nice, good general protection. But again, you're being given these antibodies, so there's no reason for your body to produce memory cells because it's not actually fighting anything. It's not initiating anything itself. It's that teach a man to fish kind of thing, where your body is just doing it for you. It's just fighting these infections for you, or the antibodies that you're getting through breast milk are just fighting the infections for you, so your body doesn't have to do anything, and so it doesn't learn. Artificially acquired passive. This one's a little tricky. This one, but again, if we have COVID to, I'm not gonna say what I was gonna say, but we have COVID to be able to say, hey, we know a little bit about serum transfers now. So for artificially acquired artificial passive immunity, this is when you are given a transfer of antibodies. Let's say, again, COVID. You are in the hospital, you are very, very sick. You didn't, your body is not producing enough antibodies. You didn't have a vaccine for it. Let's say it was early days, and because I'm sure we all understand the importance of vaccines and how they help us in these immune responses. And so you didn't have that, though. You didn't have yours. And so you're at the hospital, you're really sick. It's too late for the vaccine. The vaccine's going to take 2 weeks before it kicks in. So what they can do instead is that they take people who have recently had COVID and they take their blood, they centrifuge it out to separate the antibodies, the serum, from the actual red blood cells. They give them their red blood cells back. We don't need those. And then they take the serum that has the antibodies against COVID, and then they'll give you an injection of that. So that's the artificially acquired passive. It's, again, similar to this breast milk example, wherein you're being given antibodies, but rather than being given through breast milk, you are being given an injection of the antibodies from someone who either was recently infected or they were able to synthesize these antibodies in a lab. There's still no immune response, no antigen exposure, no memory cells produced, so it does not make you immune from that pathogen. It just gives you temporary. It's a Band-Aid, right? It's kind of probably more than a Band-Aid because it can really wipe out the virus within you, but you can still get reinfected, the next day. And because your body is not producing these antibodies, by the way, when this artificially acquired passive, you would then have to go through another transfusion to be able to have antibodies again, because these are going to get broken down pretty quickly. here's a table just outlining all of that. So make sure you read through the different types and you understand their mechanisms and results. So let's look at a couple of hypersensitivity reactions, some specifics here. So hypersensitivity, excess immune response to a normally harmless antigen that can lead to tissue damage. Can occur in almost anyone. Some do require hereditary disposition. Type 1 is you become desensitized by producing IgE antibodies in response to the presence of an antigen. This could be, let's say that you were eating something, or that you get exposed to a certain perfume, or you have a skin, well, not skin sensitivity. That one is different. But let's say, yeah, like let's do food or perfume scent allergy. During that first exposure, that antibody attaches itself to these inflammatory cells, and they start to release their components. That causes mucus production, vasodilation, bronchoconstriction, all resulting in an inflammatory response. Essentially, the body's trying to get that thing out of you, and so it's causing this overreaction of the inflammatory response, which actually ends up closing your airways and clogging you up. It can occur within seconds of an allergen exposure, and those mediators flood the body and can result in anaphylaxis, severe form of immediate reaction hypersensitivity that can be life-threatening. Antibody-dependent cytotoxic reactions. This one involves IgG, sometimes IgM. It takes 1 to 3 hours to develop. This example, transfusion reaction to mismatched blood. So something that does not happen immediately within seconds, but takes a little bit of time. Immune complex reactions, again, 1 to 3 hours to develop. IgG, an example for here is rheumatoid arthritis. This is something that my mother has. And so she has this buildup of antibodies that attack her joint cells and cause a lot of pain. Delayed reaction hypersensitivity. This is one I'm very susceptible to. I get very allergic to poison ivy. It can take a couple of days to develop. It involves T cell collection in the skin and activation. T cells and macrophages release chemicals causing eruptions and inflammation. Yes, not fun. Transplant reactions. So transplant reactions, when those are unsuccessful, is an immune response. Its transplantation is replacing a non-functional, damaged, or lost body part with one from a donor. Transplant recipient's immune system can recognize that this is something foreign, and so they do a lot of different tests to try and make sure you're a match. They look at a lot of different components, not just blood type, but organ size, the type of organ that it is, and a lot of other different markers between organs, and it depends on which organ it is. Some are easier to transplant than others. Some need to be closer matches than others. But they do all these things to try to make sure that they match as close as they can. And then a lot of times rejection can still happen. You can still have a tissue rejection reaction. One thing they do to try to help that is immunosuppressive drugs. So by suppressing the donor or the recipient's immune system, it allows the donor's organ to be integrated a little bit more, and maybe for some of those antigens to start getting replaced with self-antigens, so that way there is not as much of an immune response. That, of course, then does leave you to be more susceptible to an infection, just a normal flu, staph, MRSA infection, whatever it may be in the area. So still big risks with transplant reactions. And then autoimmunity, autoimmune disorders. This is when the immune system turns against the body or a body component, and it loses the ability to distinguish self from non-self antigens. The body starts to manufacture what we call autoantibodies, cytotoxic T cells against some of its own antigens, some of its own body cells. These can damage the body's tissues. It can happen from a viral infection, faulty T cell development, or a reaction to a non-self antigen that closely resembles a self antigen. So maybe it's just one that's like, well, this kind of looks like us, and because we had a reaction to this one, now we're really paranoid about all of these, and so the body starts attacking itself. Type 1 diabetes, diabetes mellitus, that's from an autoimmune disorder in which the beta cells are attacked and destroyed by autoantibodies. All right, thank you so much.